Hello and welcome to SAM online class. This is Dr. Subrat Kumar Mahanti. Friends, today we shall discuss about telomere and telomerase. Telomeres are the physical ends of eukaryotic chromosome. And telomerase is an enzyme that is responsible for maintenance of length of telomere by adding guanine-rich repetitive sequences. Telomere protect the ends of chromosome from DNA degradation, DNA recombination and fusion. By repressing telomerase and induced shortening of telomere provide the means to regulate the cellular lifespan. Hermann Muller and Barbara M.C. Clintock were probably the first scientists to recognize that telomere has special properties and crucial function. And the molecular sequence of telomeric DNA was first determined by Elizabeth Blackburn and Joseph Gall. The term telomere was introduced by Hermann Muller. The term telomere is derived from two Greek words. Telos means end, meros means part. Telomeres are special nucleoprotein structure that are present at the ends of eukaryotic chromosomes. The vertebrate telomeric DNA, it contains repetitive sequences. The repetitive sequences contain thymine, adenine, guanine, and guanine. This is the repetitive sequence that is present in the telomere. The telomere length of human being is considerably shorter. The telomere of human chromosome varies from 5 to 15 kilobase. And the telomere of human chromosome, it contains repetitive sequence variant of the repeat thymine, adenine, guanine and guanine. So the telomere is the physical end of chromosomes that contain the repetitive sequence thymine, adenine, guanine and guanine. This is the repetitive sequence that is present at the telomere. So friends, this is an chromosome. This is an chromosome and these are the ends of chromosome. These ends of chromosome are known as telomeres. And this is the base sequence of telomeric DNA. You see, in this telomeric DNA sequence, the repeat sequence thymine, adenine, guanine and guanine, it has been repeated. And the telomeric DNA, it terminates with a 3' overhang. The double-stranded telomeric DNA has a 3' overhang that is guanine rich. So in this diagram it is very clear that these are the ends of chromosome and these ends of chromosome are known as telomeres, and the telomeres contain repetitive sequence that is thymine, adenine, guanine and guanine. So such sequence is repeated in a telomere, and the telomeric DNA, it has a 3' overhang. This is the 3' overhang which is rich in guanine. The double-stranded telomeric DNA terminates at a 3' guanine rich overhang. This is the 3' guanine rich overhang which contains about 12 to 500 bases. Mammalian telomeric DNA has been found to fold into a lariat-like structure known as telomere loop or T-loop. This is the T-loop that is formed due to the folding of the telomeric DNA. In this T loop, the 3' overhang invert the double-stranded region and form another small loop that is known as displacement loop or D loop. The T loop acts as a protecting cap. It shields the chromosome ends from being recognized as damaged DNA. It prevents the nucleolytic degradation and inappropriate fusion of telomeres. The T loop is stabilized by a protein complex known as shelter-in. Shelter-in is a protein complex also known as telosome. It protects the telomeres and it also regulates the activity of the enzyme telomerase. The shelter-in, it is composed of 6 subunits. The 6 subunits are telomere repeat binding factor 1, telomere repeat binding factor 2, repressor activator protein 1, protection of telomere 1. Another subunit is TPP1 and the last subunit is TRF1 and TRF2 interacting nuclear protein. So these are the 6 subunits that are present in a shelter-in. Now we shall discuss about the various subunits of shelter-in. The first subunit is TRF1. TRF1 is a homodimeric protein. It binds with double-stranded region. Second subunit is TRF2. TRF2 is another protein that is structurally related to TRF1. TRF2 also bind with the double-stranded region. Both TRF1 and TRF2 participate in the replication of telomere. They also prevent the stalling of the replication fork. TRF1 and TRF2 recruit the remaining subunits of the shelter-in to the telomere. Third subunit is repressor activator protein 1. Repressor activator protein 1 interact with TRF2 and it inhibits DNA repair. Fourth subunit is protection of telomere 1. This protection of telomere 1, it binds to the single-stranded DNA. It contains OB folds and this subunit help in the formation of D loop. This subunit shelter the 3' overhang. This subunit shelter the 3' overhang and it prevents the degradation of single-stranded DNA. Fifth subunit is TPP1. TPP1 is another subunit that interact with both TIN2 and POT1. The loss of TPP1 impair the function of protection of telomere 1. Last subunit is TIN2. TIN2 means TRF1 and TRF2 interacting nuclear protein. TIN2 interact with both TRF1 and TRF2 and also with TPP1. So all these protein factors constitute the shelter-in and they play specific role in the maintenance of the telomere. Many other factors also interact directly or indirectly with the telomere. One is PIP2. PIP2 means phosphatidylinositol 4,5-bisphosphate. It also interacts with the telomere. Another protein is EST1A. EST1A is a single-stranded telomere binding protein. It is an enzyme that is coded by the SMG6 gene. This SMG6 gene resides on chromosome 17. EST1A play a role in telomere maintenance. Another enzyme is telomerase. Telomerase is another enzyme that is encoded by TNKS gene. This enzyme inhibits the binding of TRF1 to the telomere. This is about various factors that interact directly or indirectly with the telomere. Due to the inability of the conventional DNA polymerase to completely replicate the linear chromosomal DNA, and also due to additional nuclease degradation of telomere, the telomere in somatic cell progressively shorten as the cells divide. A critically short telomere lead to telomere degradation. This activates the DNA damage response pathway. So, the telomere in dividing somatic cell progressively shorten. This leads to cellular senescence or apoptosis. Apoptosis is also known as programmed cell death. This is visualized as a mechanism to limit the proliferative ability of the dividing cells. The enzyme telomerase is expressed during early embryogenesis and subsequently downregulated in most of the somatic cells. The activity of telomerase is tightly regulated both at the level of its expression and its biogenesis. Majority of the tumor cells acquire cellular immortality and stabilization of telomere length by activating a normally dormant enzyme known as telomerase. Another term is TERRA. TERRA is an abbreviation for telomere repeat containing RNA. This is the RNA sequence that is transcribed from telomere. Human TERA is transcribed from several or possibly all chromosome ends. The transcription of this RNA, it starts in the subtelomeric region and proceed up to 500 nucleotides into the pure TTAGGG tract. TERRA function along with shelter-in to inhibit the lengthening of telomere by the enzyme telomerase. Telomerase, also known as telomere terminal transferase, is an enzyme that catalyzes species-dependent addition of telomere repeat sequence to the 3' end of telomere. Telomerase is a ribonucleoprotein. It consists of a protein part and a non-protein part. The non-protein part is a ribonucleic acid. The catalytic core of telomerase consists of two main components. One component is TERT. TERT means telomerase reverse transcriptase, and other component is the telomerase RNA. The telomerase RNA serves as the template for the addition of telomere repeat sequence to the 3' end of telomere. Telomerase reverse transcriptase, it catalyzes the reverse transcription of the template RNA present in the telomerase to produce telomere sequence repeat at the 3' end of the telomere. The telomerase reverse transcriptase can catalyze the addition of telomere repeat sequence in an iterative manner. The propensity of some telomerases to add several telomere repeats without dissociation is known as repeat addition processivity. TERT, that means telomerase reverse transcriptase, it is composed of several subunits. This component possesses the canonical reverse transcriptase moiety. Other subunits are telomerase essential amino terminal domain and telomerase RNA binding domain, and also there is a carboxyl terminal extension. All these domains act together to catalyze the process of reverse transcription by utilizing the template RNA that is present in the telomerase. The TAT also contains some other subunits which are less well conserved. This is about telomerase. Now we shall discuss about certain difficulties that arise during the replication of telomere. In the double-stranded telomeric DNA, one strand is rich in guanine. That strand is known as G-rich strand, and the other strand is known as C-rich strand. The G-rich strand is replicated discontinuously in the form of Okazaki fragments. This is the Okazaki fragment. Each Okazaki fragment starts with an RNA primer. The removal of last RNA primer leaves the gap for which there is no mechanism to fill in. The C-rich strand is replicated continuously, making them blunt-ended. After this, the C-rich strand is processed and resected with the help of specific nucleases and helicases, including mammalian Apollo, to generate the 3' guanine-rich overhang, which can be folded to form the T-loop in order to ensure the stability of the telomere. During movement of the replication fork through the telomere, the fork faces several potential challenges. The challenges are G quadruplexes, heterochromatinized DNA, and T-loop. G quadruplexes is also known as G4. It is a stable intramolecular or intermolecular structure that is held by 4 guanine residue through Hoxton base pairing. During the replication of telomere, the separation of the two strands generate. During replication of the telomere, the separation of the strands expose the G-rich strand that facilitates the formation of G quadruplexes. The G quadruplexes is a stable structure that can be resolved with the help of specific protein, Bloom syndrome protein, Werner syndrome protein, replication protein A, and protection of telomere 1. All these specific proteins act together in order to resolve the G quadruplexes. Next challenge is heterochromatinized DNA. The DNA that is present in the telomere and also in the subtelomeric region is heterochromatinized. The heterochromatinized DNA is considered as a negative regulatory mechanism for lengthening of the telomere. Third is T-loop. T-loop is itself a challenge to the movement of replication fork. And during the separation of DNA strands, topological stress accumulate in the telomeric DNA. T-loop is considered as a topological barrier. The topological stress that is generated during the unwinding of the DNA strands can only be relieved by the unwinding of the T-loop so that replication of telomere can proceed in an efficient and accurate manner. In higher eukaryotes, all these issues are tackled with the help of two protein consortiums. The components of these two protein consortiums act in a coordinate manner to ensure the passage of the replication fork through the telomere. One protein consortium include various replication proteins that work at the telomere. The replication proteins include DNA polymerase alpha primase, replication of protein A, RCQ helicases, flap E endonuclease 1 and DNA 2. RCQ helicases also include Bloom syndrome protein and Werner syndrome protein. The other protein consortium include various telomeric protein that also work at the telomere. So the components of these two protein consortiums act in a coordinate manner to ensure the smooth passage of replication fork through the telomere. The replication of telomere is thought to be initiated from a replication origin that is located in the subtelomeric region. Now we shall discuss about the synthesis of leading strand and lagging strand. The leading strand is synthesized in a continuous manner. This black color strand is the C-rich strand. This C-rich strand is replicated in a continuous manner. The red color strand is the leading strand. The leading strand is synthesized in a continuous manner. So, the DNA fragment becomes blunt-ended. After this, there is an initial resection of this C strand or C-rich strand. After initial resection, there is extension of the 3' end with the help of telomerase. Telomerase catalyzes the addition of guanine-rich repeats to the 3' end of the leading strand. After extending the 3' end, there is another C strand resection and synthesis. The C strand resection help in the generation of the 3' guanine-rich overhang. After the generation of this 3' guanine-rich overhang, this DNA is folded to T-loop in order to ensure the stability of the telomere. Next, synthesis of lagging strand. This blue color strand is the parental strand. This strand is rich in guanine. This strand is replicated in a discontinuous manner in the form of Okazaki fragments in 5' to 3' direction. The leading strand is also synthesized in 5' to 3' direction. Each Okazaki fragment starts with an RNA primer. After removal of the last RNA primer, there is a gap that cannot be filled due to the absence of 3-hydroxyl group. You know that DNA polymerase is an enzyme that is primer dependent. It can add nucleotides one after one to a free 3-hydroxyl group. So there will be no further extension. So a gap is present here. After this, the enzyme telomerase will bind with the guanine-rich strand. This is the guanine-rich strand. So there is complementary base pairing between the RNA sequence. This is the RNA sequence that is present in the telomerase. This RNA sequence acts as a template for the reverse transcription into the DNA telomeric repeats with the help of enzyme telomerase. So after this base pairing, with the help of this enzyme telomerase, there is an extension of the 3' end. So after this extension, the enzyme telomerase can move further and it can add another telomeric sequence repeats. So through repeated extension and translocation, the reverse transcriptase, it can extend the 3' end in an iterative manner. So, this activity of the enzyme telomerase help in the creation of guanine-rich 3' overhang. So, there is extension and translocation several times with the help of the enzyme telomerase in order to extend the 3' end of the guanine-rich parental strand. After extension of the 3' end with the help of enzyme telomerase, there is necessity for the synthesis of the strand in place of the RNA primer. And here there is a RNA primer, and this RNA primer has been removed, and after the removal of RNA primer, this is the gap that has to be filled. This gap is filled with the help of other enzyme primase and DNA polymerase. The enzyme primase catalyze the synthesis of short RNA primer, to which the enzyme DNA polymerase can add nucleotides one after one on the basis of nucleotide sequence that is present in this template strand. After filling this gap, the nick which is present, it can be sealed with the help of enzyme DNA ligase. So, the enzyme telomerase play a very important role in the extension of telomere. Friends, the replication of telomere is somehow a complicated process. So please watch this video twice or thrice so that you can easily understand this biological process. Please share and like this video. Subscribe my YouTube channel, Sam Online Class. Thank you friends. See you again. Bye bye. Good night.